mafia, chain swinging, clang clang, and it costs a lot. Bitch, I'm always at the guala, yeah, and you are not badass beat. Keep on going till you hit the spot. Whoa, I'm a big bag hunter with the bow. She got a big bed, never drop it low. Mama called me and she happy with the grow. Never ever fall for a honey, that's a no.
Hello everyone, we are from AS120318. We are going to present our BIO270 case study today. Our topic for today is about rice plants. Rice plants produce an important staple food. It is crucial that this plant can thrive against stress such as drought and salinity. Explain several strategies to improve such stress tolerance in rice plants by giving examples.
Hi everyone, my name is Nur Atira. I'm going to talk about some interesting facts about rice plants. First of all, the scientific name of rice plants is Oryza sativa. It is commonly grown in Asia, such as Bangladesh, Vietnam, Thailand, Philippines, and Japan. It is one of the most important staple food and it feeds more than one-half of the world's population. In humid and subhumid Asia, rice plants is actually an important source of employment and income for rural people. It provides about 50 to 80% of calories for more than 3 billion Asians. And it is also listed as the most salt-sensitive cereal crop.
Thank you, Atira. Assalamualaikum. My name is Nur Aisara. I'm going to be explaining about the objective of this case study that we're going to achieve. So firstly, the objective is to identify the strategies to improve stress tolerance in rice plants. Secondly, to identify the effects of the strategies towards rice plants.
The first strategy is breeding for stress tolerance. There are two types of breeding. The first one is conventional breeding. It is also known as traditional breeding. It is a method of developing new plant varieties by selecting and crossing individuals with desirable traits. In 1939, Pokali, which is the first salt-tolerant rice cultivar in the world, are able to tolerate salt concentration for more than 0.3% in soil. And in 1985, Jiangsu Academy of Agricultural Science are able to identify even more rice cultivar, which is Baiguzi and Zuziki. The first step of conventional breeding is identify desirable traits. Breeders have to identify specific traits that they want to improve and choose breeding population that is highly variable for salinity and drought tolerance. The second step is parental selection. Breeders have to select parental plant, which is the parental lines, to serve as the starting material for the breeding process. And the third one is progeny evaluation. The resulting offspring, known as progeny, is then evaluated for salinity and drought tolerance. After evaluation, the plants that achieve the desirable traits will be selected for further breeding. After all these steps are complete, new rice plant varieties is formed. For example, CSR13, CSR10, CSR27, IR2151, and IR36. These rice varieties is currently cultivated in many countries, and there is a photo of rice variety IR36 that have been uploaded by Bharat V in 2016. Conventional breeding has obtained success in raising rice genotype with improved tolerance to salinity and droughts. The second type of breeding is molecular marker assisted selection (MAS). MAS is an indirect selection for a desired plant phenotype based on binding pattern of the linked molecular DNA markers. It is applicable for genetic improvement of plants, and Saltol is widely used in MAS breeding in rice-growing countries such as India, Vietnam, Thailand, and Bangladesh. The first step of MAS is selection of parents. Parents with divergent or contrasting origin is chosen, help to identify DNA of both parents and their segments in other generations in various recombination. The second step is isolation of DNA. Breeders need to make sure DNA can be isolated even from seedlings, and the isolated DNA is digested with specific restriction enzyme to obtain fragments of DNA. And then the DNA chloroplast produce about 40 fragments of different sizes when digested with specific enzymes. And the third but not least procedure is correlation with morphological traits. DNA markers are correlated with morphological markers. Once correlation of molecular markers is established with morphological markers, MAS is effectively used for genetic improvement of various economic traits. Some advantages of MAS is it have a very high accuracy, it is not affected by environmental conditions. That means we would have no worries about salinity problems or climate change. It is a rapid method of crop improvement, and MAS has wide application for genetic improvement of oligogenic traits, which is traits that is influenced by a few genes as compared to polygenic traits, which is the characteristic. The diagram right here shows two red flags, which is the marker 1 that is more tightly linked than 2, and the green part is called as the target gene. There are few examples of molecular markers such as random amplified polymorphic DNA, simple sequence repeats, and restriction fragment length polymorphism. That is all from me. Let's continue with the next strategy. Thank you.
Okay, so next I will be explaining about genetic engineering. So genetic engineering is also known as genetic manipulation or genetic modification. So how exactly this genetic engineering works? It is by changing a single base pair, AT or CG, deleting a region of DNA, and adding a new segment of DNA. So how actually genetic engineering is applied on rice plant? So basically it is through transgenic approach. So what is transgenic approach? It is formed by inserting a desirable stretch of DNA sequence into a plant cells and then proliferating it using plant tissue techniques. The approach has a few benefits which are resistant to biotic and abiotic stress, increased nutritional value, and factors for production of recombinant protein. How transgenic approach works in rice plant? So first, it is by overexpressing ATHB101 proteins and silencing HSP101, and the next one is knockout mutants of rice, which are OSGSK1. So I'll be explaining further about these two steps, but first let's look at the background of the two steps. Okay, first, what is actually ATHB101 and HSP101? So it is basically a group of proteins that are synthesized in response to high temperature stress. So the function of the first step, which are overexpressing ATHB101 protein and silencing HSP101, it is for impaired thermal protection and enhanced heat tolerance. The second one is repairing and aiding in the renaturation of stress damaged protein, and lastly protecting cells against the effects of the heat stress. The next is OSGSK11. So it is referred to a gene in rice which are origin sativa called cotocytin kinase 1. So the function of knockout this mutant of rice, it is for regulate thermal tolerance and enhance tolerance to various abiotic stress, including high temperature. I'll be explaining about the first technique, which are overexpressing ATHB101 protein. So first, it is by isolation of the ATHB101 genes. The ATHB101 genes can be isolated from the genome of the rice. It can be done using PCR, which are polymerase chain reaction or gene cloning. And the second step is construction of gene expression vector. So isolated ATHB101 gene, it needs to be inserted into a suitable gene expression vector, and the vector is a DNA molecule that can carry ATHB101 gene. So the third one is transformation of rice cells. So the concentrated gene expression vector containing the ATHB101 genes is introduced into rice cells, and it can be done through various methods such as agrobacterium mediated transformation or biolistic particle bombardment. So the fourth step is selection and regeneration of transgenic plants. Transformed rice cells that have successfully incorporated the ATHB101 genes are selected using selectable markers present in the gene expression vector. So by this, it allows the identification of growth of transgenic cells. The selected cells are then regenerated into whole transgenic plants. So the last step is conformation of ATHB101 overexpression. The presence and expression of the ATHB101 gene in the transgenic rice plant can be confirmed using molecular techniques such as PCR or gene expression analysis. The next one is silencing HSP101. So basically, the entire step is the same as overexpressing ATHB101 protein, but there are slightly some difference. So first, it is the same as the overexpressing ATHB101, which are identification of the HSP101 genes, and the second one is design of gene silencing construct. So once the HSP gene is identified, gene silencing construct can be designed. These constructs typically contain a fragment of the HSP101 gene that is complementary to the target of the gene sequence. So the next step, transformation of rice cells. So the third step is transformation of rice cells. It is also the same as overexpressing ATHB101, which are the gene silencing construct, are introduced into the cell of the organism using the techniques such as agrobacterium mediated transformation or biolistic particle bombardment. So then the transformed cell will contain the gene silencing construct. So the fourth step is expression of the gene silencing construct. The gene silencing construct is transcribed into RNA molecules, which then form a double-stranded RNA, which are the sRNA structure. So these dsRNA molecules can trigger a process called RNA interference, which we call RNAi, which leads to the degradation or inhibition of target HSP101 mRNA. So the last step, it is also the same as overexpressing HB101, which are verification of gene silencing. So the effectiveness of gene silencing can be confirmed by analyzing the expression levels of the HB101 genes using techniques such as quantitative PCR or gene expression analysis. So the next method is knockout mutants of rice OSGAK1. So it is actually involved disrupting or deleting the target gene's DNA sequence, resulting in the loss or reduction of its normal function. So the method that we use in this knockout mutants of rice OSGAK1 is actually CRISPR-Cas9 technology. So in this method, a gap RNA is designed to target the specific sequence of OSGSK1, and the Cas9 enzyme is used to introduce double-strand breaks at that location.
Thank you, Anis. Now I'm gonna explain about another strategies that can improve stress tolerance in rice plants, which is soil management. So what is soil management? Soil management is also known as agronomic practices or crop management techniques. This management involves various methods and approaches to optimize soil conditions, nutrientability, and water supply. This management is basically essential for the overall health and stress resilience of rice plants. Now, there are a lot of methods or techniques for this soil management, including improving soil structure, addition of organic matter, proper irrigation management, and pH regulation. So the techniques will also have some advantages, which is improved soil structure, increased organic matter, water conservation, and pH balanced. So the first technique for soil management is to improve soil structure. So what is soil structure? It is the arrangement of soil particles, which are sand, silt, clay, and organic materials, into granules, crumbs, or blocks. Soils that are well structured are typically crumbly and friable, with enough pore space to facilitate water and air movement for good root development. So why structure is important? It is important because it is essential for allowing air and water into the soil. So one of the ways to measure structure is to use the soil remote sensing and modeling. Second technique is to add organic matter. So organic matter is soil organic component made up of organic material from plants and animals, as well as material converted by microorganisms in the soil at various stages of decomposition. So how to improve organic matter? We can add carbon inputs, keeping the soil covered for a longer period of time, and also using bioeffectors which stimulate biological activities in the soil. Thirdly is to do proper irrigation management. So this technique also refers to water management that carefully control and conserve water resources to ensure optimal growth, development, and yield of rice crops. So what are the methods for these techniques? We can do continuous flooding, can provide the best growth environment for rice, and we can also alternate wetting and drying for direct seeded rice, while keeping the soil moist but not saturated. This is to avoid seeds from rotting in the soil. The last technique for soil management is to do pH regulation. So first we have to know what is the pH of rice. The pH value of rice is within the range of 6 to 7. And then why is measuring the pH of rice is important? This is because the United States Department of Agriculture has set the levels of micronutrient in rice, as well as Food Safety and Criteria Authority of India has also established criteria for iron, folic acid, zinc, vitamin B12 and E for fortification of rice. So how can we balance the pH of soil? We can do mulching that can maintain the soil's acidity, and we can also use compost that is rich in organic matter and can do better than commercial fertilizer. So now that we know the techniques for soil management, I'm going to explain the advantages that we can achieve from the techniques. So when we improve soil structure, we can improve root penetration, aeration, and water infiltration. Proper drainage decreases the possibility of waterlogging, which is harmful to rice plants. Next, when we increase organic matter, we will improve soil structure, delivers critical nutrients, and promotes good microbial activity, all of which are necessary for rice plant growth. Next, water conservation can promote sustainable agricultural practices, but also enhances crop yield, mitigates environmental impact, and ensures long-term food security. The last advantages that we can achieve from a balanced pH is the assuring nutrient availability, fostering beneficial soil organisms, preventing toxicity, increasing soil structure, disease avoidance, optimizing fertilizer efficiency, and eventually producing consistent and sustainable rice yields.
In this video, we have explained about the 3 strategies to improve stress tolerance in rice plant, which are breeding for stress tolerance, which we use conventional breeding, and molecular marker assisted selection, which are must. And the second one is genetic engineering. We apply it through transgenic approach, and the last one is soil management. So by improving stress tolerance in rice plant, we can actually reduce negative effects of stress leading to better growth and yield in crops growing in poor condition. So that is all from our team. Thank you for watching.